Okay. All right, welcome back. So, I just realized that in my previous PowerPoint, some of the writing was off the slide, or off the whiteboard in a way that you could not see it. So that's kind of tough to take notes if you can't see the notes. So what I'm going to do is I'm going to make those notes available to you, and I apologize for that. I think that issue has been fixed. I've adjusted my camera, so hopefully that shouldn't happen again. So now that we've learned about the biological species concept and reproductive isolation, we're going to apply some of these ideas to talk about how species actually arise. And there are two general processes depending on how gene flow ends up getting interrupted. And these two processes are what we call allopatric speciation or sympatric speciation. Okay, so let's talk about allopatric speciation. So allopatric roughly translates to other country. So in allopatric speciation, something physically separates a population. This could be a mountain range, this could be a lake level dropping, causing a land barrier. This could be a river changing its course, this could be a canyon. So for example, these two different species of squirrels were once the same species in a continuous population that found themselves on opposite sides of the Grand Canyon. And this splintered the two populations into subpopulations. And the idea with allopatric speciation is that once our subpopulations are separated, so subpopulations get separated, again, by some kind of physical barrier, and then this physical separation, of course, keeps them from interbreeding, and different mutations arise. In the different subpopulations. Also in the different subpopulations, natural selection, genetic drift, or sexual selection also lead to genetic differences. Cause differences between the two subpopulations. And so when these two subpopulations come back together, if they come back together, but if the two subpopulations were to meet again, they could no longer interbreed. And that's when we would say they have speciated into two different species, because they can no longer interbreed due to reproductive isolation. Which, if we're following the biological species concept, which we are, we would say, voila, we have two different species. So one thing that's kind of interesting is that founder effects can also lead to allopatric speciation, and a great example is Darwin's finches. So this original source population of finch from the South American mainland became splintered geographically into many different subpopulations when it migrated to these different islands. And these different subpopulations didn't exchange genes with each other anymore, and over time different mutations arose, and over time natural selection acted differently in the different environments of these different islands, notably leading to differences in beak morphology, but also other types of different genetic changes due to sexual selection or genetic drift or other forms of natural selection, such that when these different birds were to come into contact with each other, they could no longer interbreed. So this one species splintered into 13 or so different species due to this founder event. Now, it's fairly easy to see how allopatric speciation works, right? You know, something happens, you have a barrier, and you can't interbreed, and so genetic differences arise. But what about sympatric speciation? So sympatric roughly translates into same country. So this idea that speciation is going to be happening despite the fact that you are in the same geographic location. So sympatric speciation occurs between populations in the same geographic location. So, gosh, how the heck does that happen? Well, something has to happen to disrupt interbreeding, okay? Something has to stop interbreeding. And the three things that can possibly do this, there are three things that can possibly stop interbreeding despite being in the same area, is something called polyploidy, which I'll explain in a minute, habitat differentiation, and sexual selection. Okay, let's talk polyploidy. So polyploidy is a condition where you have more than 2 sets of chromosomes. So you, instead of being diploid, which would be the normal state for organisms, right? Being diploid, again, that means you have 2 sets of chromosomes, like we're diploid. A polyploid is triploid, or hexaploid, or pentaploid, more than 2 sets of chromosomes. This happens due to an accident in meiosis. Right, this is something that just happens, because of a mistake in meiosis. Something didn't, chromosomes somehow didn't segregate properly during meiosis 1, and so what ends up happening is you end up having a gamete that has twice as many chromosomes as it should, and what happens is that you have a progeny produced that has a different number of chromosomes than everybody else in the population, right? Everybody else in the population is diploid, and then this polyploid is triploid. Well, it can't mate with anybody else in the population, right? It won't form viable gametes, but it can mate with itself. And this is very common in plants. I know you might be thinking animals can't mate with themselves. Plants can. And it can mate with other polyploids. And you might be thinking, well, how often does that really happen? Actually, in plants, kind of a lot. So in plants, polyploidy is actually pretty common. Now for animals, the mistakes in meiosis that lead to polyploidy might be just as frequent. However, animals almost never survive polyploidy. If you're born triploid, you won't be born. Animals almost never survive this, with a couple of very rare exceptions. But in plants, it doesn't seem to be a big problem. So we have a lot of, it's very, very common in plants. And something like 80% of modern plant species are actually formed by polyploid speciation. So things you're really familiar with: oats, wheat, which I think is a hexaploid, cotton, potatoes, strawberries. Strawberries are actually octoploid. Lots of cultivated plants, either accidentally or intentionally bred by humans to be polyploid, because polyploid plants tend to be larger because they're making more protein because they have more DNA. So essentially with polyploidy, you can get speciation in one generation, because the hybrid, or not the hybrid, but the polyploid is automatically reproductively isolated from everybody else in the population. Another kind of interesting way we can get speciation despite overlap in geography, what we would call sympatry when you're in the same area, is sort of micro-differentiation of the habitat. So in habitat differentiation, we have genetic factors arising that allow some members of a population to exploit a resource not usable to the parent population. So some members of the population, I guess another way we can say this, are using the habitat differently than the parent population. And this leads to disruptive selection and eventually reproductive isolation. Okay, so that might be a little confusing. Let's hash through an example. So this darling organism is called the apple maggot fly. I know its name is as adorable as it is. And so these apple maggot flies like to live on and eat hawthorn fruits. And they also like to mate where they eat, because everything's right there. That's just how they roll. So these maggot flies live, breed, have their life on these hawthorn fruits. That is their original habitat. Now, about 200 years ago, apples were introduced into North America. And some individuals in that population, and when I say parent population, I just mean the original population. So some individuals out of that original population were like, hey, these apples are pretty awesome. And they started hanging out on the apples and eating the apples. Now, why this matters is that apples mature faster than hawthorn fruits. So this led to faster developing flies being selected for, because they want to reproduce and have their babies at the same time that the fruit is ready to eat, so that they have food for their babies. So what happened is the population, the part of the population, or if you want to call it the subpopulation, that stayed with the hawthorn fruits were maturing later in the year when the hawthorn fruits ripened, and the flies that had switched to the apples, the subpopulation on the apples, were maturing. There was pressure to mature and mate earlier in the year. So we got this disruptive selection, right, where two extreme phenotypes are favored. The phenotype that, in the hawthorns, the phenotype that matured later in the year was favored. In the apples, the phenotype that matured earlier in the year was favored. And this also meant that the hawthorn flies were not, there was a temporal isolation mechanism happening here, right? The hawthorn flies were not mating with the apple flies because they were sexually maturing at different times of the year. So despite the fact that the apple tree might be right next to the hawthorn tree, they didn't have opportunity for interbreeding. So this could lead to reproductive isolation. So the final scenario that can lead to reproductive isolation, even despite a sympatric situation, is sexual selection. Right, so this is where individuals with certain traits have higher reproductive fitness. And a great example of this can be seen looking at a study where they took two species of male fish that lived in the same lake. So we have two species of fish from same lake, okay? So they are sympatric. We have this species and this species, and as you can see, they have some different color patterns, right? This one has this very obvious kind of orange stripe or orange, you know, sort of back, and this one doesn't. This one has some orange kind of on the fins back here, this one less so. So there's some different color patterns. And under normal light, where these color patterns were very obvious, the females preferred their own species, and 100% of the time, the female for this species mated with the right male. They always picked the right male under normal light. What the researchers then did is they put the fish under a monochromatic orange light. So it kind of negated the differences in the orange patches, right? That you could no longer tell, and they looked much, much more similar. And what we saw is that the females under the orange monochromatic light mated with either male indiscriminately. They didn't care. And not only that, but their offspring were viable and fertile, meaning that female mate choice based on male coloration was the only factor of reproductive isolation. And this evolved despite the fact that they lived in the same lake. Given enough time, we would expect that other genetic differences might arise that would keep interbreeding possible. But right now, clearly, this sexual selection is what's driving it. So that is another situation where we can get reproductive isolation in a sympatric situation. So the last little topic I want to talk about is adaptive radiation. So adaptive radiation is the proliferation of a large number of species in a relatively short period of time. I should be more specific here and say it's proliferation of a large number of species from a single ancestor species in a short period of time. This can be caused by founder events, by mass extinctions, or by innovation of new traits. So what you're looking at here is founder events. Okay, so here is the founder event of what we call the Hawaiian silver swords. Which are a genus of plant we find among the different Hawaiian islands. They are descended from a single common ancestor found on the west coast of the United States known as the tarweed. I don't think, do we have a picture of the tarweed? Oh, right here. So here is the tarweed in the center here. Oops, oh, highlighter. Okay, well there's our little tarweed. And this tarweed, seeds of this tarweed made its way to the Hawaiian Islands, to the different islands, which had, of course, had very different habitats. And in a very short period of time, we had a very large number of species in this genus known as the silver swords, all with very different morphologies arising in a very short period of time from this one ancestor due to this founder event of coming to the Hawaiian Islands. We can also get we can also get adaptive radiation following mass extinction. So the Cretaceous mass extinction killed off the dinosaurs. Killed off the non-avian dinosaurs. Birds survived, but killed off, the big giant lizard looking dinosaurs we're familiar with. And when this happened, we had an adaptive radiation in the mammals. So prior to the Cretaceous mass extinction, mammals were not very diverse. They were mostly nocturnal and they were very small. But once the dinosaurs were extinct, mammals all of a sudden had all of this ecological opportunity. It's sort of like islands of opportunity opened up to them, right? And so we had this massive adaptive radiation as different organisms diversified to meet these different newly available ecological opportunities. We also can have adaptive radiation following some kind of major evolutionary innovation. And the example I'll use here is the innovation of the evolution of flowers. So the evolution of flowers led to a whole new way of reproducing. And this led to a very quick adaptation of numerous species. All of a sudden, numerous species started popping up, each adapting to perhaps different pollinator species or evolving different traits to increase the attraction of those pollinator species, or maybe having differences in the shape of their flowers so they could more accurately place their pollen on these different pollinator species. And we just got this boom of lots and lots of different flowering plant species happening relatively quickly because of this innovative new trait. Okay, that's the end of the line for chapter 22. As always, feel free to email me with any questions you have, and I will talk to you later.